Now let's read this passage again, a passage from environmental science, and we will read the passage first. We have 4 questions in this passage. The questions are on the right hand side and the passage is on the left hand side. So let's read the passage. It says cultivation of a single crop on a given tract of land leads eventually to decreased yields. One reason for this is that harmful bacterial phytopathogens, organisms parasitic on plant hosts, increase in the soil surrounding plant roots. So again, you have lots of technicality and you have to be very careful, because don't try to unnecessarily go into the details. The author says that cultivation of a single crop can decrease yields, and the reason is given. And then he says the reason. So let's not check. Lots of names are there, technical names. So the author says in the first sentence that cultivation of a single crop leads to decreased yields because of some reason, and the problem can be cured by crop rotation. However, even if crops are not rotated, the severity of diseases brought on by such phytopathogens often decreases after a number of years, as the microbial population of the soil changes and the soil becomes suppressive to those diseases. So the author says even if you don't rotate the crop, the problem after several years decreases in intensity. Why? Because the soil becomes suppressive. While there may be many reasons for this phenomenon, it is clear that levels of certain bacteria such as Pseudomonas, antagonistic to a harmful phytopathogens, are greater in suppressive than in non-suppressive soil. So the author says that one of the reasons why after a long time the crop becomes, the severity of the harmful bacteria decreases because of the level of certain bacteria that are antagonistic to harmful pathogens. That means Pseudomonas is your friend and the phytopathogen is not your friend. This suggests that the presence of such bacteria suppresses phytopathogens. So phytopathogens cause harm, and if you maintain, if you carry on growing a single crop without rotation, gradually you will have Pseudomonas that are antagonistic to phytopathogens. There is now considerable experimental support for this view. Wheat yield increases 27%. Wheat yield increases of 27% have been obtained in field trials by treatment of wheat seeds with fluorescent Pseudomonas. Similar treatment of sugar beets, cotton. So the first paragraph is a long one, but if you start breaking down the relationship, you will realize that the author initially talks about single crop, and if you grow a single crop, you will have phytopathogens, and if you keep on growing the single crop for a long time, the phytopathogens increases initially, but later on the soil becomes suppressive. And why it becomes suppressive? Because of the presence of Pseudomonas. So basically Pseudomonas is beneficial to us, phytopathogens are not beneficial to us, and Pseudomonas are enemies of phytopathogens. And there is an experiment which was done, and the experiment was done in which the wheat seeds were wheat seeds were treated with Pseudomonas, and there was increase in the yields. So we know that Pseudomonas is beneficial to us and it is harmful to phytopathogens, and phytopathogens are dangerous. They multiply when you grow a single crop, but when you rotate the crop, they don't multiply. So this is what we have. Again, the problem is that once you have this kind of technical passages, you have to be very careful with what you read, otherwise it's going to be a problem for you. So you have to be slow and steady. You can't read the passage with the same speed always. So there has to be a choice. For example, when you're writing the paper, when you come across passages that are often from your expertise, in the sense that you are familiar with the content, you can solve those passages first and then move on to those passages whose contents are less familiar to you. So let's read the second paragraph. It says these improvements in crop yields through application of Pseudomonas suggest that agriculture could benefit from the use of bacteria genetically altered for specific purpose. So which bacteria like Pseudomonas, they can be useful. For example, a form of phytopathogen altered to remove its harmful properties could be released into the environment in quantities favorable to its competing with and eventually excluding the harmful normal strains. Some experiments suggest that deliberately releasing altered non-pathogenic Pseudomonas syringe could crowd out the non-altered variety that cause frost damage. Opponents of such research have objected that the deliberate and large-scale release of genetically altered mild bacteria might have deleterious result. Deleterious means harmful. So here the author has given the example of Pseudomonas, which is a beneficial bacteria. And then the author goes on to say that how we can use genetically altered bacteria for our own benefits. And then there are opponents who say that such kind of things might have deleterious effects. Proponents argue that this particular strain is altered only by the removal of the gene responsible for the strain's propensity to cause frost damage, thereby rendering it safer than the phytopathogen from which it was derived. So the author says that, the proponents say that only one gene has been altered, and that gene was responsible for frost damage. So if you alter that gene, if you remove that gene, the bacteria is not harmful. That is what the proponents say, and the opponents have their own objection. And then further in the paragraph, we have some proponents have gone further and suggest that genetic alteration techniques could create organisms with totally new combination of desirable traits. So it says some proponents have gone further and suggest that genetic alteration techniques could create organisms with totally new combinations of desirable traits not found in nature. For example, genes responsible for production of insecticidal compounds have been transposed from other bacteria into Pseudomonas that colonize corn roots. Experiments of this kind are difficult. So again, this para is about proponents of genetically altered bacteria. See, whenever you have too much of technicality, you just check what exactly is the para doing broadly. Experiments of this kind are difficult and require great care. Such bacteria are developed in highly artificial environments and may not compete well with natural soil bacteria. Nevertheless, proponents contend that the prospects for improved agriculture through such methods seem excellent. These prospects lead many to hope that current efforts to assess the risks of deliberate release of altered microorganisms will successfully answer the concerns of opponents and create a climate in which such research. So, I have difficulty understanding this paragraph, but I know that the proponents of genetically altered bacteria have given lots of argument, and I can come to this part of the passage if there is a question on this, on the proponents and so on and so forth. And the paragraph immediately above that talks about Pseudomonas and how Pseudomonas has been argued by both opponents and proponents. How the opponents and proponents have argued in favor or against Pseudomonas. And the first paragraph we have. So we know that the author seems to be focusing on Pseudomonas, but the focus is not Pseudomonas. The focus is that genetically altered bacteria. That is what the focus is. He starts with Pseudomonas and phytopathogen being the harmful bacteria and Pseudomonas being the beneficial bacteria, and then he gradually moves from the beneficial bacteria to give us some kind of exposure to the genetically altered stuff, right? Genetically altered bacteria, and then he keeps on discussing it. So this is what we have, and let's go ahead and take the questions, and let's see what the questions ask. So the question says, which of the following best summarizes the main idea? So again, you have the main idea question, which is again one of the typical questions that you might come across as the very first question. You have the choice of either taking this later on and solving the other questions first. Of course, there is a choice, because sometimes in the very first reading you may not be able to solve questions on which of the following is best summarizing the passage. So you can take second, third, fourth first, and then come on to first. But let's take the first one, because we are not here solving the paper in the examination, but we are here taking it for practice. So it says recent field experiments with genetically altered Pseudomonas bacteria have shown that releasing genetically altered bacteria into environment would not involve any significant danger. So the author says recent field experiments with genetically altered Pseudomonas bacteria have shown that releasing genetically altered bacteria into environment would not involve any significant danger. Looks good. We can shortlist this. But let's look at the other choices. Encouraged by current research, advocates of agricultural use of genetically altered bacteria are optimistic. But the problem is, in option B, the author uses the word genetically altered bacteria. And option A focuses on genetically altered Pseudomonas bacteria. So we have to question whether is the passage only about Pseudomonas, or is the passage broadly talking about genetically altered bacteria. Now there is definitely optimism. The optimism is there and the opponents are wary. So there is no problem with this choice B. Choice B is complete. Why? Because it says that encouraged by current research, advocates are optimistic. At the same time, opponents are wary. Wary means cautious. So it looks a good choice. The advocates are optimistic, the opponents are wary, and they are optimistic pertaining to what? Pertaining to the genetically altered bacteria. Not necessarily. Pseudomonas is just an example of genetically altered bacteria. You cannot simply say that the passage as a whole discusses about Pseudomonas. Pseudomonas is just a small example of genetically altered bacteria. So the author is discussing everything by bringing the example of Pseudomonas, but that is not the focus of the passage. And secondly, the proponents and the opponents are giving their arguments. That has not come in option A. So A is not as good a choice as B is. Then you go to C. Current research indicates that adding genetically altered Pseudomonas bacteria to the soil surrounding crop plants will have many beneficial effects. Again, the problem is you're focusing too narrowly on the Pseudomonas syringae, and secondly, you're not mentioning about the optimism of the advocates and the pessimism of the opponents. So again, incomplete and partial option. Too much of a narrow focus and not covering everything. And D says genetic alteration of a number of, you're not altering the harmful phytopathogens, you're altering the Pseudomonas. So again, it goes off the track. Genetic alteration of bacteria has been successful in highly artificial laboratory, but opponents of such research have argued that these techniques are unlikely to produce organisms. Is it only about opponents? And secondly, genetic alteration of bacteria has been successful in highly artificial laboratory conditions, but opponents of such research have argued that these techniques are unlikely to produce organisms that are able to survive in natural environments. Is this the argument that they would not survive in natural environment? The argument is not about this. The argument is about dangers. It might have lots of side effects. So that's the reason why they are cautious. The argument of the opponents is not from this point of view, survival in the natural environment. So again, you're moving away from the core stuff. So E also goes out. So let's go and take the second one. It says the author discusses naturally occurring Pseudomonas fluorescens bacteria in the first paragraph. So we know that Pseudomonas has come as an excuse for a bigger picture. So he says prove that increases in the level of such bacteria in the soil are the sole cause of soil. See, soil Pseudomonas definitely once it increases, it is the cause of the soil suppressivity. But can we say it's the sole cause? See, understand, the author is not arguing whether this is the sole cause or there is no other cause. You are taking the focus away from the purpose to something else, as if the purpose of the author is to just discuss that this is the sole cause and there is no other cause. But the author's motive is not to show that this is the sole cause and there is no other cause. The motive is that Pseudomonas is one example of one example of a beneficial bacteria, and therefore genetically altered bacteria can be beneficial as a whole. That is the purpose. So A is not the intention of the author. And then B says explain why yields increased after wheat fields were sprayed with altered. The wheat fields were not sprayed. It was not the wheat fields. Understand, you are again distorting. This distortion is so common. It says here that wheat seeds. It says here, look at this sentence. It says the treatment of wheat seeds with Pseudomonas. So the wheat seeds were treated with Pseudomonas, and in the passage, in the option, you say that wheat fields were sprayed with Pseudomonas. The seeds were treated and you say that the fields were sprayed. How? This is what happens in the examination. You mark choices which are not matching with what is given in the passage. So to someone who is, let's say, not very much good with RC, wheat field spraying with Pseudomonas is equal to wheat seed being treated with Pseudomonas. That's not the same thing. C says detail the chemical. Where do you see the chemical processes? We don't see any such chemical processes and there is no detailing. Provide background information to support the argument that research into the agricultural use of genetically altered bacteria would be fruitful. Now, why is the author saying that this is the background information? Background information of what? Background information that the way Pseudomonas is beneficial, genetically altered bacteria might be beneficial. Why? Because the author has given more examples and the author is in fact taking tacitly supporting. He says here, these improvements in crop yields through the application of suggest that agriculture could benefit. Now, this is whose opinion is this? This is the author's opinion. And then he gives the example. And then he says opponents. So the author is giving his own opinion, which is very much favorable. And therefore, we can say that author is definitely giving background information through Pseudomonas that agriculture can benefit. So D looks a good choice. E says argue that crop rotation is unnecessary since disease is brought on by phytomonas diminution delivery. Is he arguing that crop rotation should not be done? He has not said that. He said that if you grow a single crop, the single crop develops phytopathogens, and because of that, the yield decreases. And if you rotate, there's a solution for that. And if you don't rotate, even without rotation, after several years, the severity of the phytopathogens decreases. So he has not said that there should be no crop rotation. Replace crop rotation with something else. That is not the argument. In fact, crop rotation would be a good idea. Should you wait for the Pseudomonas to? There are two ways of looking at it. Either you spray it with artificially, genetically and modified bacteria, or you have crop rotation. The author has not said crop rotation is not good. Nowhere has he anywhere suggested or implied that crop rotation is not good. So E goes out. So the best answer is D, because the background information is coming to us in the form of Pseudomonas fluorescens, and that is precisely what the author wants to say. That's the way Pseudomonas comes and benefits the soil. The genetically altered bacteria might be beneficial for us. So it's a good thing. Question number 3. It can be inferred from the author's discussion of Pseudomonas bacteria that which of the following would be true of crops impervious to parasitical organisms. So impervious basically means not affected. So if you are impervious to something, that means basically you're not affected by it. So if the crops are not affected by bacteria, that means Pseudomonas will not have any impact on them. Isn't it? So you are adding Pseudomonas because Pseudomonas will benefit. But if the crops are impervious to parasitical, see, Pseudomonas is a parasitical organism. Are we getting this point? The author has clearly said that Pseudomonas is a parasitical organism. If you scroll up here, it says here that if you want to say whether it's a parasitical organism or not, in fact, it is a parasitical. It's a parasite. So the author has said that organisms parasite. So phytopathogen is an organism's parasite. And then he says that, however, if crops are not rotated, the severity of the disease brought on by such pathogens, while there may be many reasons for this, it is clearly that certain levels of bacteria, such as harmful bacteria is a phytopathogen, and Pseudomonas is a beneficial bacteria. So they both are organisms parasite. I mean, they are parasitic. So and the reason why phytopathogen is harmful because the reason why Pseudomonas is beneficial is because Pseudomonas attacks phytopathogens. So one bacteria is beneficial, the other is harmful. So they both are actually parasitics. They are both are parasites. So look at this. It can be inferred from others' discussion that Pseudomonas. So it says here it can be inferred from the author's discussion of Pseudomonas bacteria that which of the following would be true of crops impervious to parasitical organisms. So if the crops are impervious to parasitical organisms, that means Pseudomonas would not have much impact on them. And similarly, phytopathogens would not have much impact on them. Because phytopathogens are parasitic, they won't have any effect. So in fact, the yield would not decrease because of phytopathogens, and the yield would not increase because of Pseudomonas. So you can directly say that their yield would not be likely to be improved because they're insensitive, they're impervious. And Pseudomonas fluorescens bacteria would be absent from the soil. They might be present, but the effect may not be there. Understand this, the Pseudomonas might be present in the soil, but they may not have the impact because the crops are impervious. The imperviousity of the crop has nothing to do with the presence or the absence of the Pseudomonas. They would crowd out and eventually exclude other crop plants. Nowhere said again. So they all go out. Option C is the best choice. You spray Pseudomonas, you have greater yield. If the crop is impervious, the spraying would not matter much. Why? Because it will not have any result. Simple. You have to be straight point to the point. There is no need to go to beat by the bush. Right? The fourth question. Which one of the following, if true, would most seriously weaken the proponent's argument regarding the safety of using altered Pseudomonas syringae bacteria to control frost damage? So we know that something regarding frost damage has come in the paragraph. So let's go and check where exactly that has come. and it has come somewhere in the second last paragraph. That is what I remember. So definitely not at the start. So the frost damage word has come here, right? So it says here that Pseudomonas syringae could crowd out the non-altered variety that causes frost damage. So Pseudomonas syringae crowds out the non-altered variety that causes frost damage. Opponents of such research have objected that the deliberate and large-scale release of genetically altered might have deleterious results. Proponents, on the other hand, argued that this particular strain is altered only by the removal of the gene responsible for the strain's propensity to cause. Propensity means inclination. So the point here is that some experiments suggest that deliberately releasing altered non-pathogenic Pseudomonas syringae could crowd out altered variety that causes frost damage. So what is the meaning here? Altered non-pathogenic. That means Pseudomonas is of two kinds now. One is altered and the other is non-altered. So what happens? That if you deliberately release the altered non-pathogenic Pseudomonas, they will throw out the non-altered variety that causes frost damage. So as a result of spraying the altered Pseudomonas, the non-altered is getting lost, and because of that the frost damage will not happen, because the non-altered is causing frost damage. The altered is not causing frost damage. Why? Because the alteration has been done only by the removal of the gene responsible for the frost damage. So if that gene is removed, the frost damage will not happen. So you have to go and weaken this argument. The question says, which of the following, if true, would most seriously weaken the proponent's argument regarding the safety of altered Pseudomonas? The altered Pseudomonas is safe because, as per the proponent, the altered Pseudomonas is safe. Why? Because the gene that causes frost damage has been removed. But you have to weaken that. So you have to find a flaw in that idea of removal of the gene. And D says, often genes whose presence is responsible for one harmful characteristic must be present in order to prevent other harmful characteristics. So the gene that is causing frost damage is responsible for harmful characteristic, but it must be present at the same time to prevent other harmful characteristic. So there is a gene, this gene is causing one harmful characteristic, at the same time it is preventing other harmful characteristic. So if you remove the gene, definitely frost damage will not happen. But the other two things which were harmful would not be prevented, and therefore there would be a loss. And therefore D is definitely weakening the argument. So the argument is pertaining to the benefit. A says Pseudomonas are primitive. No problem, they can still be useful, and the argument is still valid. The altered bacteria are derived from a strain that is, they can be derived from anywhere, but the altered is not harmful. As long as it is not harmful, it's a good thing. So current genetic engineering techniques permit a large-scale commercial. So if they are permitting large-scale commercial, but it's a good idea. As long as it is not harmful, you can go for large-scale production. So this is positive. So how we are weakening the? E is talking positively about the altered Pseudomonas. So D is the only choice that is weakening, and weakening with the relevant point, and it is the best choice. So overall, it's a technical passage, but people understand once you have a grip on what exactly you are reading. You'll have, see, the grip basically means understanding the broader elements. Then you can crack the passage.